The Department of Urology here at the University Hospital in Freiburg is one of the busiest departments that we have in Germany. We have around 60 inpatient beds and treat more than 20,000 outpatients per year. Our focus is clearly on oncology and specifically on robotic surgery, but we cover a very wide spectrum ranging from andrology, stones, incontinence, and we have a very big focus on BPH as well. My understanding of somebody who has a chair in urology is to take care of research. So we have a wide area of research that we cover. We do clinical trials, we treat many patients, but we also do a lot of translational research. And that translational research is about bringing inventions from bench to bedside.
We have different collaborations, starting with basic scientists in the field of medical engineering, then of course with other clinical partners from other institutions, academic and non-academic institutions, and also from abroad. So we've got a lot of international collaboration and of course industrial partners allowing us to come from the clinical concept or prototype to a study to be able to apply this technology in the treatment scenario.
UCare was an initiative that we started around 5 years ago. Ian Frew was the initiator, and we quickly gathered a group of people that were interested in doing research on bladder cancer.
We're trying to understand the whole process of bladder cancer formation by combining the expertise of molecular biologists, of protein biochemists, of pathologists, of people working with cell and mouse models, as well as with human samples. And we're really trying to understand why epigenetics is so important in bladder cancer and how we can use this knowledge to better treat these tumors that patients have. Our idea is that for every patient who develops bladder cancer, they will have the tumor's genome and epigenome sequenced so that we can really understand what is driving that tumor and be able to tailor then the therapy to that particular combination of mutations and epigenetic alterations.
Finally, last year we received a large grant from the German Research Foundation, and we're very happy to be able to start this research on epigenetics in bladder cancer.
PSMA is the abbreviation for the prostate-specific membrane antigen. This is a protein which is on the surface of prostate cancer cells. And about 20 years ago, we developed some of the first antibodies which specifically bind to PSMA on the surface of prostate cancer cells. And when the antibodies bind, they are taken up into the cell. And with our antibodies, we can now transport, for example, toxins into the prostate cancer cells so that the toxins can exert their cytotoxic activity in the cells and kill them. On the other side, we can also use these antibodies to guide patients' own immune cells to the cancer cells so that the immune cells can be activated and attack the cancer cells. So it's a kind of immunotherapeutic approach.
So what we try to do is to use PSMA in detecting cancer where it shouldn't be. For example, after radical prostatectomy, we try to locate PSMA positive cells within the human body and then try to eradicate those PSMA positive cells to be able to make sure that no tumor is left within the body after prostatectomy.
KMT9 is an epigenetic enzyme, and it's very important to control and regulate prostate tumor proliferation and migration. And since it's an enzyme, we can inhibit those enzymes, and consequently we developed a highly specific KMT9 inhibitor molecule, which we are now testing in animal models, and we hope that we will bring this molecule in the next 3 to 5 years into a phase 1 clinical trial. It involves actually all areas of state-of-the-art research. So we do crystallography, we do a lot of bioinformatics, next generation sequencing, also KI-assisted drug development, and of course we do general molecular biology and also a lot of mouse work, both in mice and rats.
So BPH is a very large focus. We treat around 700 patients with laser enucleation every year. We have a large group that works on laser techniques, and we try to improve techniques using a laser in every aspect.
We have different collaboration partners, national and international partners from different research institutes or also from the industry to bring our diagnostic or therapeutic approaches into the patient.
Our research activities have led to some patents and technologies. For instance, a new laser that is able to spot the target and distinguish between stones and tissue to protect organs while doing stone fragmentation. There is another project on sensor-based system for patient monitoring after surgical interventions. Also a new laser system that has been improved and now is in the clinical use, and many more.
So whether it is with CAR T cells, whether it is with bispecific antibodies, in all these settings we're trying to improve the efficacy and also try to maintain a tolerable safety for our patients in order to prolong their lives, but also to improve quality of life, which is what it boils down to at the end, the most important part.